So we proceed with question number 9. So just as a reminder, before we proceed, kindly ensure that you like, share and subscribe to my channel. So name important components of nucleotides. So the major important components of nucleotides are nitrogenous base, phosphate group and pentose sugar. So you will get the detailed information from my notes. Search for Tom Waswa. Don't forget to search for Tom Waswa and like and share. Also subscribe to my channel. So what is the importance of nucleotides? That is the second part of our question. What is the importance of nucleotides? So for you to recall the importance of nucleotides, you need to remember first of all nucleotides themselves. So the first nucleotide that we meet is ATP. You know ATP is an important energy currency of a cell. We meet ATP in many biochemical reactions. So the first one being glycolysis. So in glycolysis, ATP is consumed in the first stages. Then it's also produced in the latter stages. We also meet ATP in electron transport system, where it is normally produced. So I won't go into details about the same. So then we have cyclic AMP and cyclic GMP, which are normally used as intracellular messengers. Then also this ATP, GTP, UDP, CoA are used as coenzymes. So we meet these molecules in various biochemical reactions. So you will meet them if you are just beginning of biochemistry, you will meet most of them. Then we have deoxyribonucleotides. Deoxyribonucleotides. These are used in DNA repair and replication. Then we have cyclic AMP and cyclic GMP, which are used as intracellular messengers. So once again, ATP, GTP, UTP, FAD, NAD, and CoA act as coenzymes. Then another important point is deoxyribonucleotides are required for DNA replication and repair. Then ATP is a currency of free energy for all cellular activities like cellular contraction, biosynthesis of molecules, and transfer reactions. Then intracellular messengers, I've just said that cyclic AMP, cyclic GMP are involved in peptide hormone action. Then GTP is used in protein synthesis. So you'll meet this one in my notes. That is on nucleic acids or genetics. So we go to question number 10. So question number 10. Using specific examples, distinguish between essential and non-essential amino acids. So from the word essential, you need to know that essential means important. So essential amino acids are those amino acids that are required in large quantities in our bodies. So they are required to be given in foods. Non-essential amino acids are those amino acids that are not required in the food because our bodies can synthesize them. So once again, essential amino acids are not synthesized in the human body. They are provided in the diet, whereas non-essential amino acids are synthesized by the body and need not to be provided in the diet. So we have said using specific examples. So we want to use examples. We want to see how to distinguish between essential and non-essential amino acids using examples. So let's say you are in an exam room or you are revising for your exam, and today is Friday and let's say your exam is on Monday. So how do you easily remember these amino acids? Well, it is very easy. I got this mnemonic from the internet. You can create your own mnemonics. That is methods of remembering things. So for you to remember essential amino acids, there is this mnemonic which says try this VIP mole. try this VIP mol. So tri stands for tryptophan. Tryptophan. Before I go there, first of all, you need to know that here we have just one amino acid. Here we have 2 amino acids. Here we have 3 amino acids, and here we have 4 amino acids. So when you take 1 plus 2, you get 3, plus 3 you get 6, plus 4 you get 10. So we have only 1 amino acid here. So tri means tryptophan. Tryptophan. This here we have 2. So the first one is threonine. The first one is threonine. And the second one is histidine. You can see the initial of histidine. So you can get histidine from this. Then we have VIP. V stands for valine. There are 3. So we have valine here. Valine. We have isoleucine. I stands for isoleucine. P stands for proline. Valine, isoleucine, and proline. Then let's go to mol. The first M stands for methionine. Then we have arginine. We have leucine and also lysine. So you need to pay attention on this A because several of them begin with A. We have alanine, aspargine, aspartate, but then the one that is required here is arginine. So arginine is an important amino acid, an essential amino acid. So we begin try this VIP mol. So from the word VIP will help you to remember that they are important. They are essential amino acids. So using this mnemonic, you can recall the essential amino acids, and therefore you can categorize them into essential, then non-essential. So let's say you are in an exam and you've been given a group of amino acids, and you are being told to put aside essential and non-essential amino acids. So this is the only way you can easily distinguish them. So let's begin. Essential are tryptophan from the group that we've been given. The first one is tryptophan, then followed by threonine, then histidine, then we have valine, we have isoleucine, we have proline, that means we are here. Then we go to methionine, then arginine, then leucine and lysine. Then that means that the remaining amino acids can be placed here. Then an essential amino acid. So I can see cysteine. We can place cysteine here. We can place alanine here. Aspargine, aspartate. Glutamine, glutamate, glycine, tyrosine, phenylalanine, and valine. Phenylalanine and valine. Phenylalanine and valine. I think I have remembered something there. So this can help you to distinguish them. Please, if you have anything about the comments, come in the comment section and comment. I really appreciate that. So we proceed. We go to question number 12. Describe the process of glycolysis. Then which steps are irreversible? So the major irreversible steps of glycolysis are step number 1, step number 3, and step number 9. Those are the major irreversible steps. Those are the irreversible steps. So we want to describe glycolysis, but how can you easily remember the glycolysis, the steps in glycolysis? So to easily remember the steps in glycolysis, let us start by naming the molecules. Let's remind ourselves on how to remember the molecules of glycolysis. So we begin with our substrates. So in my mnemonic, I have named the first six substrates. So these ones, after naming them, then naming the enzymes, you will easily state all of the molecules and the enzymes involved in glycolysis. So some of the students have been asking me, how can I easily remember glycolysis? So I want to begin by, first of all, giving you a mnemonic on how to remember the molecules of glycolysis. So the first mnemonic stands for the substrates. So it goes as follows: Gladys, gladly, fried fruits on damaged gas, on damaged gas. So gl, the first GL means you are dealing with glucose. Then FR stands for fructose, fructose, okay? This is glucose, glucose, fructose, fructose. On, this one is just like that, it is there. Then D means dihydroxyacetone phosphate. Then we have gas. This one stands for glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So after stating those molecules, so these are 6 molecules, you will easily remember the other molecules, and I'm going to give you a hint on how to easily remember the other molecules. So that is the first mnemonic. Then the second mnemonic stands for enzymes. You say, you remember about someone called Helen. Helen does what we call inspection of pictures along training ground. So you say Helen inspects pictures along training ground. Along training ground. That is the first mnemonic on enzymes. Then the second part of the mnemonic of the enzymes, I want to remember the following. I want you to remember that praying mutants enjoy painting. Praying mutants, praying mutants, praying mutants enjoy painting, enjoy painting. Praying mutants enjoy painting. So if you have those mnemonics, you can easily remember glycolysis. So let me show you how. So let's start by naming our molecules. We've said that the first 6 of the molecules are as follows. So the first one is glucose. The first one is glucose. Let me use black for the substrates. So in the first step, we are converting glucose into glucose 6-phosphate. Remember, the first step is irreversible. Glucose 6-phosphate. Allow me to write phosphate as P. The next step is reversible. So from glucose 6-phosphate, we form what we call fructose 6-phosphate. Then from fructose 6-phosphate, we go to, this one stands for glucose. This one is glucose 6-phosphate, fructose 6-phosphate. We go to another fructose. So it is fructose 1,6-bis phosphate, and that is step number 3. It is irreversible. So fructose 1,6-bis phosphate. Now we have 2 phosphate groups. So from glucose we go to glucose 6-phosphate, then we go to fructose 6-phosphate, then fructose 1,6-bis phosphate. Then from there we say that on does not have any meaning. So this is number 1, number 2, number 3. We go to number 4. Step number 4. Step number 4, these steps are reversible. So fructose 1,6-bis phosphate. Now we are forming dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So I want us to be writing it like this: dihydroxyacetone phosphate plus glyceraldehyde 3-phosphate. Then from the naming of those molecules, you also need to know that this is another step where we are going to convert another dihydroxyacetone phosphate into another molecule of glyceraldehyde 3-phosphate. So this is another important step. It is a reversible step. So after that, I want us to go to enzymes, back to enzymes. I said Helen inspects pictures along training ground. Then praying mutants enjoy painting. So Helen stands for hexokinase. Then inspects. I stands for isomerase. Isomerase. Pictures. P stands for phosphorylation. We are carrying out phosphorylation reaction, so it is a phosphofructokinase. Then A stands for Al stands for aldolase. Al stands for aldolase enzyme. Then Tr stands for triose phosphate isomerase enzyme. Then we continue. I want us to continue from there because this is what we have just formed. So we start naming the enzymes here. So our first enzyme is a hexokinase. So the role of a kinase enzyme is to transfer a phosphate group from one molecule onto another. So in this case, we know that a phosphate group is involved. So we are consuming ATP. So we are going to convert our ATP into ADP. And we transfer a phosphate group to glucose to form glucose 6-phosphate. Then the next step, we are going to convert glucose 6-phosphate into fructose 6-phosphate. So glucose 6-phosphate and fructose 6-phosphate are isomers. So our enzyme is isomerase. And also from our mnemonic, we know that this one, we have said that this one stands for isomerase. Then we go to the next step. We are converting fructose 6-phosphate to fructose 1,6-bis phosphate. So this is a fructose 6-phosphate. Remember the name of our enzyme starts with P. So this is phosphorylation. P stands for phosphorylation reaction. So our enzyme will be phosphofructokinase. Phosphofructokinase enzyme. So that means we are adding another phosphate group. By the way, when you look at fructose 6-phosphate and fructose 1,6-bis phosphate, you will realize that you are adding a phosphate group on fructose at position number 1. So in this case, we are going to convert fructose 6-phosphate into fructose 1,6-bis phosphate. That means we are going to consume our ATP. So our ATP is going to be converted into ADP. Then we continue. Fructose 1,6-bis phosphate is converted into 2 different isomers, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So the breaking down of fructose 1,6-bis phosphate being a bilateral molecule, we call this one lysis. So our enzyme that is involved in lysis is aldolase. So this one stands for aldolase. So our enzyme is aldolase. Our enzyme is aldolase. Then when you look at dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, you realize that these are two isomers. So our enzyme must be an isomerase. So remember TR. So what is it? These molecules contain 3 carbon atoms. So our isomerase is a triose phosphate isomerase. It is carrying out isomerization of triose phosphates. So triose, so that is triose phosphate isomerase. That is the name of our enzyme. Then the next step that I have not mentioned here, let's see how to remember it. This is glyceraldehyde 3-phosphate. And our steps are as follows. That is step number 1, 2, 3. Step number 4. Step number 5. We convert this one into the other one. Then step number 6. Step number 6 is the one that follows. We want to add another phosphate group. So this is the only step that you need to remember in your head. That step number 6 involves the following: addition of an inorganic phosphate in our structure, and also the production of NADH. So we convert our NAD into NADH. You know the importance of NADH. This one is a high energy electron carrier molecule that is used in the transfer of electrons in the electron transport system. So glyceraldehyde 3-phosphate, we want to form the next molecule. When you look at this, it will help you to remember that you are adding a phosphate group on glyceraldehyde 3-phosphate. So we'll have 3 phosphate groups on our glycerol molecule. So we shall have 1,3-bisphosphoglycerate. So that is our next molecule. So that is 1,3-bisphosphoglycerate. So we go to the next step. Now we have our phosphate groups. This is our step number 6. Step number 7, we have 3 phosphate groups on our molecule. So another thing that we also need to remember on in glycolysis is that step number 7 and the last step are ATP synthesis steps. So 7 and 9 are ATP synthesis steps. So here we are going to transfer a phosphate group from 1,3-bisphosphoglycerate onto ADP. We are using blue, sorry. Onto ADP to form ATP. So we are going to convert our ADP by interacting with inorganic phosphate to form ATP. So our inorganic phosphate will come from 1,3-bisphosphoglycerate. So now that we've removed one phosphate group from position number 1 of 1,3-bisphosphoglycerate, we expect to remain with 3-phosphoglycerate molecule. So 3-phosphoglycerate. Don't forget about the enzymes. So our enzyme in this case it starts with the G. So this is our glyceraldehyde 3-phosphate. So we are carrying out the hydrogenation reaction. So our enzyme is supposed to be glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. I want to write it like that. Then our enzyme in this case, we are synthesizing what? We are synthesizing our ATP, our first ATP, and we are transferring a phosphate group from a phosphate group from a phosphoglycerate onto ATP, onto ADP to form ATP. So our enzyme will be phosphoglycerate kinase. Phosphoglycerate kinase. So our enzyme is phosphoglycerate kinase. Then see how the next step will be very easy. So this is number 7, this is step number 8. But remember, these steps are reversible. These steps are reversible. Don't forget that. So we go to step number 8. Go to step number 8. So we are going to shift the phosphate group from position number 3. This is 3-phosphoglycerate. I hope you do not confuse with my 8 here. We want to transfer a phosphate group from position 3 to position number 2. So to form 2-phosphoglycerate, 2-phosphoglycerate, 2-phosphoglycerate. So we are just shifting the position of a phosphate group. That means that this molecule and these molecules are isomers. So a special type of isomerase enzyme that is involved in the transfer of a phosphate group from one position to another is known as mutase. So our enzyme is mutase. So we are already in our second mnemonic. We say that praying, remember this is phosphoglycerate kinase, mutants stand for mutase enzyme. Then enjoy means inolase. So we go to step number 9. We say that step number 1, 3, and step number 9 are irreversible. So our enzyme here is inolase, from enjoy, inolase. Our enzyme here is called inolase. So inolase helps in the conversion of 2-phosphoglycerate to phosphoenol pyruvate. So let's see what it does. First of all, how do you know that it has helped in conversion of 2-phosphoglycerate to phosphoenol pyruvate? So inolase aids in a condensation reaction. This is step number 9. It aids in condensation reaction where we have the formation of water molecule. The formation of a water molecule. So it carries out dehydration reaction whereby it dehydrates the 2-phosphoglycerate molecule into phosphoenol pyruvate. So we go to the next step, and this is step number 9. Remember, step 9 is irreversible. We go to step number 10. In step number 10, we are going to convert phosphoenol pyruvate into pyruvate. Phosphoenol pyruvate into pyruvate. Phosphoenol pyruvate into pyruvate. So, in your knowledge, what do you think is the name of our enzyme? We have transferred a phosphate group somewhere else because we are remaining with a molecule that does not have a phosphate group. So our enzyme must be a kinase because it is being involved in phosphorylation. This process is called phosphorylation. So our enzyme is pyruvate kinase. Our enzyme is pyruvate kinase. Pyruvate kinase. So you need to know that this step involves ATP synthesis. So it involves the transfer of a phosphate group from it involves the combination of ADP and inorganic phosphate to form ATP. So at this juncture, we have managed to highlight the steps in glycolysis. We have described the glycolysis. So the first step involves phosphorylation reaction where glucose is phosphorylated by ATP to form glucose 6-phosphate by an enzyme known as hexokinase, that is helen. Then next, glucose 6-phosphate is converted into fructose 6-phosphate by a process by an enzyme known as isomerase. I've just shown you all how to remember these enzymes of glycolysis and the molecules of glycolysis. So by this, you know it. If you want to understand glycolysis in terms of chemistry, I have uploaded the notes on glycolysis in terms of chemistry. So we can go to the next part of the question.